Good evening, everyone. My name is Krishna Prasad. I am a stem cell researcher, and today I'm going to present a very interesting paper that came recently in a reputed publication about the claiming about the effectiveness of a homeopathy. So let's start the presentation. So you might have seen this news cutting in one of the social media platforms like Facebook or Telegram, saying that a new study sheds light on the effectiveness of a homeopathy medicine. And I have focused on the major findings of it. A study fills gap of lack of experimental and scientific evidence behind the action and non-clarity of the healing mechanisms of the homeopathic medicine. So you might be familiar that homeopathy is a pseudoscience. It's not just a pseudoscience. To me, homeopathy is just a tooth fairy science. Tooth fairy science is nothing but the study of things that do not exist. To me, that's just a it's not even a parody of medicine. It's just a joke, and it's a very dangerous kind of alternative medicine that harms many patients every year, and even pushing some of the patients into the worse condition and even death as well. So this news cutting that came in the Times of India, which is a reputable newspaper, and it has massive amount of circulations. And I think this news gives a false information to the readers of all languages. And I think that it's very important to actually debunk what is actually the study claims, and is there anything that we can actually use from the study for science. So before going into the science and the paper itself, let me give you a brief introduction about brief introduction about the study. So imagine that you have a colorful dye, maybe like of a bluish tinge or a cyan or something like that. Then you take the dye, then you dissolve it in a fixed amount of water. Then it dissolve it properly. It gives a very homogeneous solution of a blue color. You call it the mother tincture or the main solution or something like that. Then you are going to dilute the solution. So you are already familiar with. You don't have to go to a school, or you don't have to go to university, or you don't have to do a PhD to understand that. I think almost all the kids, when they used to play in their childhood, knows that if you dilute a colorful solution, the intensity of the color will reduce over time. So imagine that you are doing an experiment like this, that you are diluting a colorful solution, and then somehow you are measuring the intensity of each solution, and you are writing a thesis and publishing it in a reputed journal, and your conclusion is like this: the dye is good for watching Netflix. Then what will be your reaction then? Definitely you'll be saying that your experiments doesn't coincide with your conclusions. Your conclusion is diametrically opposite, or it has not even any connection with the experiments or the theoretical questions that you wanted to explore, right? This is exactly what happened in this homeopathic study. We will go to that. This study called Unraveling the Low Frequency Triggered Electromagnetic Signatures in Potentized Homeopathic Medicine. It was published in Elsevier. To talk about Elsevier is a very reputed publishing company, and this journal, Material Science and Engineering, it has an impact factor of about 3.4. We know that when we discuss about a scientific study, the impact factor of the journal in which it is published is very important. Most of the time, it is very important to know the actual impact factor of the study. We don't consider studies that are published in low impact factor journals, like journals that are having an impact factor of less than 1. So the question is what impact they are actually making if they are having a impact factor of less than 1. But this is a mediocre journal of having a impact factor of around 3.4, and it is coming from a reputed publishing company. So the question is how this bogus study, or like this what we call it's a pseudo scientific study, how a pseudo scientific study can publish in a reputed journal like this. Okay, let's go and explore what they did. What they're actually trying to do in this study is that they wanted to create a device, or create a novel device to actually categorize the potencies of the homeopathic medicines. So that's what their intention was, because they mentioned in the introduction of the paper that after a dilution of a 12C, it is impossible to actually find any molecules of the medicinal molecule. So they wanted to make a device, a novel device, so that it can categorize the homeopathic medicines even in low potency. So if we take a for example, if you take a homeopathic medicine of like 30C or something, then put it into this machine or this device, then it gives the results that, oh, it is of this potency. So that's what they wanted to actually create. And in the conclusion, they are mentioning that the homeopathic medicines have a indigenous electromagnetic signatures of their own. And these electromagnetic signatures, when the homeopathic medicine is taken, induces or like activates the vital energy in the human body. And that's how it heals any ailments. That's what what's their claim is. It's not even science fiction to me. It is like just a piece of joke. Okay, so this is the instrument they have made to measure this extraordinary science. When looking at this picture, these device may not be very familiar to you, because even the authors claim that it is a very novel instrument. So there is no chance that you have seen it, but you have seen this in your life. Not only you have seen this instrument, you have experienced with this instrument in your life, but in a different form. If I show the picture of that instrument, you can definitely definitely identify that instrument. And that instrument is nothing else, a metal detector. You might have used a metal detector in your life, or you might have gone through a metal detector door, and you have seen this thing. And this is exactly what you have just seen before. I will explain that in detail. So in order to understand a bit about what the authors have done in this study, it's necessary to understand a bit about the how a metal detector actually works. So I just wanted to remind you some of the very simple and very fundamental things that you have studied in school in 8th or 9th or 10th standard, that if you have electricity, you can create magnetism. And if you have magnetism, then you can create electricity. So the example for the first one is, if you connect a copper wire to a battery and wind it around an iron rod, then it can deflect a compass. This is a very classic home-based experiment we have studied in school, or maybe we have studied in school, or maybe we have done this experiment in home. It's one of the what we can say, the science experiments, the very amazing and fascinating science experiments that we can do in our home. It's a very cheap one. I think anybody can do this. So what this experiment tells is that when you connect the copper wires to a battery, it is deflecting a compass. That means the wire is now producing a magnetic field. The magnetic field is now interacting with the compass. The compass, before the interaction with the magnetic field, it is now it was aligning with the magnetic field of the earth. Now a nearby magnetic field interacts with it, and it deflects the compass. That's basically what happens. It basically means that the wire is producing a magnetic field. And I said magnetism can also produce electricity. The better example of that one is the dynamo in a bicycle. You know that if you when you run a bicycle, the wheel inside the dynamo rotates, and there's a magnet inside the dynamo. And when the cycle is running, the dynamo rotates, and it changes the magnetic field. And the change in the magnetic field induces a form of induces a current, and when the current flows through a lamp, the lamp of the cycle glows. So this is a very simple thing you might have always seen in your life. Even this dynamo technology is actually used in almost all forms of current production, like windmills, hydroelectric powers, and a lot of stuffs. So the takeaway is like electricity can produce magnetism, and magnetism can produce electricity. Now coming back to the functioning and or the working of the metal detector. So the metal detector has a coil which is called the center coil. So what happens in the center coil is that when you press a button, current flows to the center coil, and the center coil produces a magnetic field. So when that magnetic field interacts with a target or a metal, this magnetic field induces a current in the target, and that current in the target produces a magnetic field in the target, and that magnetic field, when interacts with another coil in the metal detector called the receiver coil, induces a current in the receiver coil, and that current, when flows into a loudspeaker, it makes that beep 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 sound that we are familiar with the metal detector. So this is exactly what you see in this novel experimental setup. So it is not basically a novel experimental setup at all. It is just what do you call it? It's like a modified version of a metal detector or altered form of a metal detector. The only difference here is that they have changed the loudspeaker. They have changed the loudspeaker and attached a spectrum analyzer to it. So what is the spectrum analyzer is doing, or what is a spectrum analyzer? You know that a loudspeaker, what a loudspeaker is doing, a loudspeaker is converting the electrical energy into sound energy, and the spectrum analyzer, what it does is it converts the electrical signals into a form of light energy, in the form of some graphs or something, so that you can visualize the amplitudes and the shape of the electrical signal that is coming out from the experiment. So that's what the spectrum analyzer is doing. Then you have a function generator here. So what is basically a function generator? Function generator is nothing but the input that you are giving into the experiment. So with the help of the function generator, you can create electrical signals with the different frequencies of your interest. That's a function of a function generator. So let me walk you through the experimental setup. You have a function generator to give the electrical inputs of a varying frequency and all. And when the current flows from the function generator to a coil that you can see in the figure, it's called the excitation coil, which is labeled there. And in the excitation coil, then you can see a greenish a cylindrical tube where you fill the homeopathic medicines. And above that, then you have a sensing probe, which senses the magnetic field that is produced. So when you put a current from the function generator to the excitation coil, it produces a magnetic field, and the magnetic field interacts with the homeopathic medicines in the cylindrical tube, and that homeopathic medicines, according to the Faraday's laws of induction, produces a magnetic field, and that magnetic field interacts with the sensing probe, and that sensing probe gives the analyze in the spectrum analyzer, and it gives the shape of the electrical signal that is coming out of the sensing probe. So it is that simple. So in this study, they have used metal based medicines. In homeopathic medicines, there are a lot of comedy stuffs. You have medicinal plants that are used as homeopathic medicines. Even cockroach they use as medicinal plants, as medicines. They use even the moonlight. It's called Luna. There's a medicine in homeopathy called Luna, which is nothing but the moonlight. So homeopathy is, that's what I said. It's a tooth fairy science. It's a study of something that doesn't exist. So in this study, what they took is like metal-based homeopathic medicines. For example, ferrum metallicum. Ferrum metallicum is nothing but a homeopathic medicine made up of iron, and they have taken the ferrum metallicum sample Then they have a diluted into 6 potencies: 1x, 2x, 3x, 4x, 5x, and 6x. And they have measured the voltage that is coming out of the sensing probe when the function generator throws a current to the electrical excitation coil. That's the basic idea of this experiment. And these are the results that's coming out of the spectrum analyzer. What I wanted to tell you, or what I wanted you to think, is this: have you able to get any information from this graph that you do not know before? That's something that I wanted to ask you. Like, uh, whatever that is shown in this graph, it is already familiar to a kid. I mean, like, those who have played with the colored waters, they actually knows this. That's what they are showing in this graph. Just look at the graph. The y-axis plots the microvoltages that is produced by the sense, that is detected by the sensing probe, and in the x-axis there it is showing the frequency of the current that is given as the input. So they have made the graph at a frequency of a 300 hertz, and they have measured the voltage and produced this graph. You can see from this graph that 1x has the high voltage, and 2x has lesser voltage than 1x, and 3x has lesser than 2x, and 4x has lesser than 3x, and 5x and 6x are nearly overlapping each other. So this is what I said. We know that 1x, 2x, 3x, 4x, 5x, and 6x means the sample is getting diluted each time. So the 2x sample has a less concentration than the 1x, 3x has less concentration than 2x, and 6x has less concentration than 5x. That's a very basic fact everyone knows, and that's what just we are saying here. Like, when the potency decreases, there is not much material to give a secondary magnetic induction, and there will be lesser voltage in the sensing probe. This is a very, 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 very simple thing that you can understand without doing an actual experiment. This is what this is the reason why I said this is a very stupid and very funny study, but accidentally and unfortunately published in a very reputed journal. And the figure B is nothing but figure A, but an enlarged image of a figure A showing the overlapped 5x and 6x. And coming back to the next graph of what they were actually trying to do is that they increased the frequency of the input to a 4.7 kilohertz, that is 4700 hertz. Why did they do that? Because they wanted to get a higher resolution of the 5x and 6x, because in the previous image those 5x and 6x were almost overlapping each other. So they wanted to just now separate them, or give a better resolution of that. In order to do that, they have increased the frequency of the current, and when the frequency of the current is increased, the magnetic field in the secondary magnetic field, the strength of the secondary magnetic field will be increased. That's a basic science that we studied in school, and they have did that, and they have got a separation now for the 5x and the 6x. And I don't actually understand why they did that, because they are not gaining any useful new information in this graph from the previous graph. As I said, like, if you increase the frequency of the current, then the strength of the magnetic field will definitely be increased, and then you will be able to see a resolution. You will be able to see the graph getting separated. And there is nothing big here. There is nothing actually useful to publish here, but they have published it. And this graph, can you see this graph? It is plotting the voltage and the concentration in the y-axis. This, to me, this graph is not much different from the previous graphs, but it in a very different shape. The previous graphs were in a very strange shape, and now you have kind of a line graph. I don't understand, like, whether these people who actually did this research were coming from school, or like they were actual scientists or not, because To me, these all graphs are the same thing, telling the same story again and again in very different forms. So look at the first graph. First graph, a graph, the y-axis is voltage and the x-axis is the concentration. And you already know that if the concentration is decreasing, then the voltage will decrease, because I already explained the reason for that. When the concentration decreases, there won't be any actual material in the sample to give a secondary magnetic field. And if there is a very less amount of magnetic field, then there will be less amount of a voltage. That's shown in the graph. Actually, I don't need to do this experiment to understand that. I can basically think, because if you know the basics of physics, or the fundamentals and the principles of physics, it's more predictable than medicine or biology. So actually, I don't need to do this experiment to understand this graph. So in the newspaper, I have seen that they have spent 2 years, yeah, 2 valuable years to do this experiment and to get published in this journal. So this is the quality of the scientific studies, especially the studies that are happening in alternative medicines or the complementary medicines, that's happening in the country as well as outside. So it's more of a joke. There is nothing useful in it. It's a complete waste of time, and money. That we should understand. That we should understand and think about it. And the graph B that you are seeing here, it's nothing but the graph A. I don't understand why they are making the same graph again and again and again and again in the whole paper. Here in the x-axis, they are plotting the differences of the concentration, and in the y-axis, they are plotting the differences of the voltages. The same graph A, they have done some very basic operations to the function, and then they have plotted the graph. I don't think even a 10th standard student will do this. It is that much of a joke, because why you need to actually plot two graphs saying the same thing with the different x-axis and y-axis. And they have actually done some more experiments in it to actually measure the concentrations of these chemicals, as well as to figure out or to categorize these homeopathic medicines with its conventional technologies like Raman spectroscopy, impedance analyzer, FTIR analysis, and all those things. And they are just irrelevant because they show a lot of mistakes, like either their machines were not calibrated or the data was manipulated. That's just what I understood from the paper, because it doesn't make any sense. Because what they were actually trying to say is that their machine, the device, the novel machine that he has seen now, the metal detector, works better than the conventional machines like the Raman spectroscopy and FTIR and all. Because when they check these homeopathic medicines of varying potencies from 1x to 6x, these conventional systems were not able to detect the iron in it or the medicines in it, but their futuristic Captain American type of device is actually able to detect up to 6x. The most funny thing that I forgot to mention here is that what I expected from this study was that, okay, they mentioned in the introduction that after 12C, it won't be able to detect any form of a medicinal molecule in their sample. That we know. It's not only, it's not just us, even like the Samuel Hahnemann, who actually devised the homeopathic system of medicine. He actually knows that, because he was a very famous chemist. He was a brilliant chemist at that time. So he knows that, like after 12C, you will not have any, not even one molecule of the medicinal drug in that sample. So what I was expecting that they will be using high potencies like 12C, 30C, 200C or 100C or 500C and all those things, because when the corona came, they were actually giving arsenicum album 30C, which they were claiming that it is an immune booster. It protects you from the infection of a coronavirus, and you don't actually have to take vaccines and all. And that was the 30C. So I expected that they will be analyzing these stuffs like 30C and all, because that is actually the real homeopathic stuff. Like that's the thing that we all always wanted to see. Like, that I was actually waiting. Like I was waiting for Avatar 2 for 10 years. When I first read the title of this paper, I was expecting that I was going to see Avatar 2. But what they did is that they stopped the experiment at 6x, and showed me a graph that even I think a 6th standard student or a 6-year kid who is playing with the colored water actually knows. And we'll come back to the conclusion. The conclusion says that that's the most funny thing. It has no connection to whatever that we have discussed so far. However, the finding from our indigenously developed experimental setup confirmed that each homeopathic medicine has its own electromagnetic signature under excitation frequency. That is not that is not even a correct a scientific sentence, because to have an electromagnetic signature for each sample, that is actually against the basic facts of science. Like, you will detect the differences, individual differences in each molecule or each sample, like the changes in the amplitude detected by the sensing probe and all. But you will now never able to detect a signal that is entirely different, that can be considered as a signature of each potency. That is, this sentence that I just read and highlighted here is not even a scientific sentence. It is a scientific sentence, and it just contradicts all the principles that we know from electromagnetism and physics. And the other one that I have highlighted, the study confirms that homeopathic medicines have electromagnetic energy even when the medicine is in very small concentrations. That is a very misleading and unscientific, and I would say that tooth fairy, and also yeah, not even a science fiction based sentence, because homeopathic medicines doesn't have electromagnetic energy associated with it, even in small concentrations, because we know that electromagnetic energy is nothing but the energy that is associated with the electromagnetic wave. It is not an energy that is associated with the what we can say, it's associated with the homeopathic medicine or not. That is just a pure, or what we can, it's a bullshit statement. I wonder how these studies are getting published in a high impact factor journal, not a high impact factor journal, in a very reputed journal. And this study actually came from CSIR, from a CSIR in Bhopal. CSIR is one of the group of best institutions in India that is doing some very serious work. And they have published a crap, and they're saying that the study confirms the homeopathic medicines have electromagnetic energy associated with the associated in low concentrations. That is not even a science. I don't think that a student that attends a 10th standard class and studies everything that being taught there will say this. So that's very strange. This is the pathetic condition of the alternative medical research system, and the alternative medicine itself in India and around the globe. And they have said in the conclusion that they have been effective in treating various ailments in patients by reviving the vital natural energy to find diseases. I have spent a good amount of time reading this crap, probably more than 5-6 hours, to really looking for anything serious in it. Unfortunately, I couldn't detect anything that is good there. It is just a crap paper. It's like a tooth fairy science paper, and they are making LKG graphs in it, nursery graphs in it, and they're saying a sentence in the conclusion that is not even science, that it is effective in treating various ailments in patients by reviving the natural energy to fight diseases. And a vital energy is a concept that's been overthrown for more than 200 years ago. in the scientific field. There was a time in science that time we believed that each biological entities have a form of energy associated with it called vital energy, and those molecules that is associated with a biological system can never be reproduced in a lab or cannot be synthesized in a lab. And that was overthrown. The entire idea was overthrown when Frederick Wöhler actually synthesized the urea in the lab, and showing that the urea, a biomolecule that can be produced without the help of any biological entity at the time. And th- like what we can understood from this statement is that like homeopathy is still in that time, and they don't even accept what happened 200 years or 300 years before, and they have never moved or developed from that single position, and writing still those fancy idiotic statements that are not even science. And the most important thing is that when a paper like this get published in a reputed journal like elsewhere, uh, there are some students that always go to homeopathic medicines and homeopathic universities and all, and to them it's an inspiration. They are students, they don't actually know how to read a research paper, to how to interpret it, how to critically analyze and analyze all those things. They think that okay, it is true, it is published in a big journal, our medical system actually works, it is discovering things that even the large hadron collider can't even discover. So they give them a false hope, like these people are giving false hope to the patients, and these research publications are giving false hope to these young students and pushing them to a career that is just completely useless and dangerous in my opinion as well. So anyway, thank you very much for listening, and I'm signing out.